Artificial photosynthesis transforms carbon dioxide into liquefiable fuels. Chemists at the University of Illinois have successfully produced fuels using water, carbon dioxide and visible light through artificial photosynthesis. By converting carbon dioxide into more complex molecules like propane, green energy technology is now one step closer to using excess CO2 to store solar energy, in the form of chemical bonds, for use when the sun is not shining and in times of peak demand. Plants use sunlight to drive chemical reactions between water and CO2 to create and store solar energy in the form of energy-dense glucose. In the new study, the researchers developed an artificial process that uses the same green light portion of the visible light spectrum used by plants during natural photosynthesis to convert CO2 and water into fuel, in conjunction with electron-rich gold nanoparticles that serve as a catalyst. The new findings are published in the journal Nature Communications. The goal here is to produce complex, liquefiable hydrocarbons from excess CO2 and other sustainable resources such as sunlight, said Prashant Jain, a chemistry professor and co-author of the study. Liquid fuels are ideal because they are easier, safer and more economical to transport than gas and, because they are made from long-chain molecules, contain more bonds, meaning they pack energy more densely. In Jain's lab, Singju Yu, a postdoctoral researcher and first author of the study, uses metal catalysts to absorb green light and transfer electrons and protons needed for chemical reactions between CO2 and water, filling the role of the pigment chlorophyll in natural photosynthesis. Gold nanoparticles work particularly well as a catalyst, Jain said, because their surfaces interact favorably with the CO2 molecules, are efficient at absorbing light and do not break down or degrade like other metals that can tarnish easily. There are several ways in which the energy stored in bonds of the hydrocarbon fuel is freed. However, the easy conventional method of combustion ends up producing more CO2, which is counterproductive to the notion of harvesting and storing solar energy in the first place, Jain said. There are other, more unconventional potential uses from the hydrocarbons created from this process, he said. They could be used to power fuel cells for producing electrical current and voltage. There are labs across the world trying to figure out how the hydrocarbon to electricity conversion can be conducted efficiently, Jain said. As exciting as the development of this CO2 to liquid fuel may be for green energy technology, the researchers acknowledge that Jain's artificial photosynthesis process is nowhere near as efficient as it is in plants. We need to learn how to tune the catalyst to increase the efficiency of the chemical reactions, he said. Then we can start the hard work of determining how to go about scaling up the process. And, like any unconventional energy technology, there will be many economic feasibility questions to be answered, as well. The Energy and Biosciences Institute, through the EBI Shell program, supported this research. Geometry of an electron determined for the first time. Physicists at the University of Basel are able to show for the first time how a single electron looks in an artificial atom. A newly developed method enables them to show the probability of an electron being present in a space. This allows improved control of electron spins, which could serve as the smallest information unit in a future quantum computer. The experiments were published in Physical Review Letters and the related theory in Physical Review B. The spin of an electron is a promising candidate for use as the smallest information unit, qubit, of a quantum computer. Controlling and switching this spin or coupling it with other spins is a challenge on which numerous research groups worldwide are working. The stability of a single spin and the entanglement of various spins depends, among other things, on the geometry of the electrons, which previously had been impossible to determine experimentally. Only possible in artificial atoms. Scientists in the teams headed by Professors Dominic Zumbel and Daniel Loss from the Department of Physics and the Swiss Nanoscience Institute at the University of Basel have now developed a method by which they can spatially determine the geometry of electrons in quantum dots. A quantum dot is a potential trap which allows to confine free electrons in an area which is about 1,000 times larger than a natural atom. Because the trapped electrons behave similar to electrons bound to an atom, quantum dots are also known as artificial atoms. The electron is held in the quantum dot by electric fields. However, it moves within the space and, with different probabilities corresponding to a wave function, remains in certain locations within its confinement. Charge distribution sheds light. The scientists use spectroscopic measurements to determine the energy levels in the quantum dot and study the behavior of these levels in magnetic fields of varying strength and orientation. Based on their theoretical model, it is possible to determine the electron's probability density and thus its wave function with a precision on the sub-nanometer scale. To put it simply, we can use this method to show what an electron looks like for the first time, explains Loss. Better understanding and optimization. The researchers, who work closely with colleagues in Japan, Slovakia and the US, thus gain a better understanding of the correlation between the geometry of electrons and the electron spin, which should be stable for as long as possible and quickly switchable for use as a qubit. We are able to not only map the shape and orientation of the electron, but also control the wave function according to the configuration of the applied electric fields. This gives us the opportunity to optimize control of the spins in a very targeted manner, says Zumbel. The spatial orientation of the electrons also plays a role in the entanglement of several spins. Similarly to the binding of two atoms to a molecule, the wave functions of two electrons must lie on one plane for successful entanglement. With the aid of the developed method, numerous earlier studies can be better understood and the performance of spin qubits can be further optimized in the future. Scientists uncover a trove of genes that could hold key to how humans evolved. Researchers at the Donnelly Center in Toronto have found that dozens of genes, previously thought to have similar roles across different organisms, are in fact unique to humans and could help explain how our species came to exist. These genes code for a class of proteins known as transcription factors, or TFs, which control gene activity. TFs recognize specific snippets of the DNA code called motifs, and use them as landing sites to bind the DNA and turn genes on or off. Previous research had suggested that TFs which look similar across different organisms also bind similar motifs, even in species as diverse as fruit flies and humans. But a new study from Professor Timothy Hughes' lab, at the Donnelly Center for Cellular and Biomolecular Research, shows that this is not always the case. Writing in the journal Nature Genetics, the researchers describe a new computational method which allowed them to more accurately predict motif sequences each TF binds in many different species. The findings reveal that some subclasses of TFs are much more functionally diverse than previously thought. Even between closely related species, there's a non-negligible portion of TFs that are likely to bind new sequences, says Sam Lambert, former graduate student in Hughes' lab who did most of the work on the paper and has since moved to the University of Cambridge for a postdoctoral stint. This means they are likely to have novel functions by regulating different genes, which may be important for species differences, he says. Even between chimps and humans, whose genomes are 99% identical, there are dozens of TFs which recognize diverse motifs between the two species in a way that would affect expression of hundreds of different genes. We think these molecular differences could be driving some of the differences between chimps and humans, says Lambert, who won the Jennifer Dorrington Graduate Research Award for outstanding doctoral research at U of T's Faculty of Medicine. To reanalyze motif sequences, Lambert developed new software which looks for structural similarities between the TF's DNA binding regions that relate to their ability to bind the same or different DNA motifs. If two TFs, from different species, have a similar composition of amino acids, building blocks of proteins, they probably bind similar motifs. But unlike older methods, which compare these regions as a whole, Lambert's automatically assigns greater value to those amino acids, a fraction of the entire region, which directly contact the DNA. In this case, two TFs may look similar overall, but if they differ in the position of these key amino acids, they are more likely to bind different motifs. When Lambert compared all TFs across different species and matched to all available motif sequence data, he found that many human TFs recognize different sequences, and therefore regulate different genes, than versions of the same proteins in other animals. The finding contradicts earlier research, which stated that almost all of human and fruit fly TFs bind the same motif sequences, and is a call for caution to scientists hoping to draw insights about human TFs by only studying their counterparts in simpler organisms. There is this idea that has persevered, which is that the TFs bind almost identical motifs between humans and fruit flies, says Hughes, who is also a professor in U of T's Department of Molecular Genetics and Fellow of the Canadian Institute for Advanced Research. And while there are many examples where these proteins are functionally conserved, this is by no means to the extent that has been accepted. As for TFs that have unique human roles, these belong to the rapidly evolving class of so-called C2H2 zinc finger TFs, named for zinc ion containing finger-like protrusions, with which they bind the DNA. Their role remains an open question, but it is known that organisms with more diverse TFs also have more cell types, which can come together in novel ways to build more complicated bodies. Hughes is excited about a tantalizing possibility that some of these zinc finger TFs could be responsible for the unique features of human physiology and anatomy, our immune system and the brain, which are the most complex among animals. Another concerns sexual dimorphism, countless visible, and often less obvious, differences between sexes that guide mate selection, decisions that have an immediate impact on reproductive success, and can also have profound impact on physiology in the long term. The peacock's tail or facial hair in men are classic examples of such features. Almost nobody in human genetics studies the molecular basis of sexual dimorphism, yet these are features that all human beings see in each other and that we are all fascinated with, says Hughes. I'm tempted to spend the last half of my career working on this, if I can figure out how to do it. The research was funded by grants from the Canadian Institutes of Health Research, the National Science and Engineering Research Council, and the U.S. National Institutes of Health. Hughes also holds the Bills Chair of Medical Research at the University of Toronto. Nature inspires a novel new form of computing. Using light, McMaster researchers have developed a simple and highly novel form of computing by shining patterned bands of light and shadow through different facets of a polymer cube and reading the combined results that emerge. The material in the cube reads and reacts intuitively to the light in much the same way a plant would turn to the sun, or a cuttlefish would change the color of its skin. The researchers are graduate students in chemistry supervised by Kalaichal Saravanamuttu, an associate professor of chemistry and chemical biology whose lab focuses on ideas inspired by natural biological systems. The researchers were able to use their new process to perform simple addition and subtraction questions. These are autonomous materials that respond to stimuli and do intelligent operations, says Saravanamuttu. We're very excited to be able to do addition and subtraction this way, and we are thinking of ways to do other computational functions. The researchers' work, published today in the journal Nature Communications, represents a completely new form of computing, one they say holds the promise of complex and useful functions yet to be imagined, possibly organized along the structures of neural networks. The form of computing is highly localized, needs no power source and operates completely within the visible spectrum. The technology is part of a branch of chemistry called nonlinear dynamics, and uses materials designed and manufactured to produce specific reactions to light. A researcher shines layered stripes of light through the top and sides of a tiny, glass case holding the amber-colored polymer, itself roughly the size of a die used in a board game. The polymer starts as a liquid and transforms to a gel in reaction to the light. A neutral carrier beam passes through the cube from the back, toward a camera that reads the results, as refracted by the material in the cube, whose components form spontaneously into thousands of filaments that react to the patterns of light to produce a new three-dimensional pattern that expresses the outcome. We don't want to compete with existing computing technologies, says co-author Faria Mahmood, a master's student in chemistry. We're trying to build materials with more intelligent, sophisticated responses. Big energy savings for tiny machines inside all of us are trillions of tiny molecular nanomachines that perform a variety of tasks necessary to keep us alive. In a groundbreaking study, a team led by SFU physics professor David Civic demonstrated for the first time a strategy for manipulating these machines to maximize efficiency and conserve energy. The breakthrough could have ramifications across a number of fields, including creating more efficient computer chips and solar cells for energy generation. Nanomachines are small, really small, a few billionths of a meter wide, in fact. They're also fast and capable of performing intricate tasks, everything from moving materials around a cell, building and breaking down molecules, and processing and expressing genetic information. The machines can perform these tasks while consuming remarkably little energy, so a theory that predicts energetic efficiency helps us understand how these microscopic machines function and what goes wrong when they break down, Civic says. In the lab, Civic's experimental collaborators manipulated a DNA hairpin, whose folding and unfolding mimics the mechanical motion of more complicated molecular machines. As predicted by Civic's theory, they found that maximum efficiency and minimal energy loss occurred if they pulled rapidly on the hairpin when it was folded but slowly when it was on the verge of unfolding. Stephen Large, an SFU physics graduate student and co-first author on the paper, explains that DNA hairpins and nanomachines are so tiny and floppy that they are constantly jostled by violent collisions with surrounding molecules. Letting the jostling unfold the hairpin for you is an energy and time saver, Large says. Civic thinks the next step is to apply the theory to learn how to drive a molecular machine through its operational cycle, while reducing the energy required to do that. So, what is the benefit from making nanomachines more efficient? Civic says that potential applications could be game-changing in a variety of areas. Uses could include designing more efficient computer chips and computer memory, reducing power requirements and the heat they emit, making better renewable energy materials for processes like artificial photosynthesis, increasing the energy harvested from the sun, and improving the autonomy of biomolecular machines for biotech applications like drug delivery.